Hi Mark, what are we doing today?
Well, we're going to extract DNA from this leaf. So it's been growing, it's come from a plant in the greenhouses. We've grown it here. We're going to put it in this tube and then we put it in the liquid nitrogen so it freezes, and then we grind it up with this and all the cells are going to break apart, and that means our DNA can come out. So we're going to use these big forceps. Okay, so there's some nitrogen in that tube now. You'll see it's subliminating.
It's bubbling away.
And then, being careful not to get my hands too close to it, we're going to put this pestle in. And you can probably even hear it crunching. Up with a nice powder in the bottom of our tube, and we're just going to add the buffer, so I'll do that next, and then we're going to put it in the incubator. Okay, and then we're going to put that in the incubator and we're going to wait for all the cells to fall apart, and that'll take about an hour. So samples have been in the incubator for an hour, but you'll see it's all mixed in with the bits of leaf mixed within the solution. So we're going to spin it very fast in the centrifuge, and that's going to bring all the green tissue down to the bottom, and we're going to use the clear liquid that comes from the top.
Everything is done now. And we see separate layers.
Alright, so next step, we've got to add a chemical called chloroform, and that's going to break up all the bits in our tissue that we've ground up that we don't want. So all the protein, all the fats and things like that, is going to break those up and separate them from our DNA. So this is our hazardous chemical, which is why we're in the fume hood. And you'll see it makes the different layers. So what we're going to do, the chloroform is going to be at the bottom and our DNA is going to be in the bit at the top. So we're going to mix it for 10 minutes, then we're going to use the centrifuge again, and it's going to separate the different layers that we want. Okay, so what we're hoping for is separate different layers, and yep, that's what we get. So this has a chloroform layer. This is the chemical we just added, and you see it's very green. In the middle we've got lots of bits floating around, so that's bits of protein and bits of lipids, and on the top it should be nice and clear, and this layer will have all our DNA in it. Next step, we're going to transfer the top layer of those tubes into a fresh tube, and then we add an alcohol, and the alcohol is going to make the DNA come out of solution so that we can actually see it and we can spin it down in the centrifuge. So a bit more pipetting. The way I do this is I usually look from the side so I can see what I'm doing. Sample 1. And sample 2. Okay. And then we just add the alcohol. Sample 1. Sample 2. Okay. And then they're going to go in the freezer for 1 hour to get them nice and cold and make the DNA come out of solution. Right, so what we want to see is nice clear liquid with a little bit of white at the bottom of the tube. And there we go. There's a little bit of white at the bottom of the tube. So that will be all our DNA. Just check the other one. Yep, that one's worked as well with a lot more DNA. So there we go, we've made our DNA.